we have a very exciting lineup of speakers for you who will be talking about ways in which they are using technology to tackle problems in healthcare and climate change and digital inclusion. So to kick us off, I am excited to introduce Kexin Xu, who is a Ph.D. candidate at the University of Chicago. I'm very sorry, at the University of Oxford. And Kexin is a graduate from Imperial College, and she is also the winner of the prestigious Clarendon Scholarship. And she will be talking to us about her research that she started when she was an EquiTech Scholar in 2022 at the intersection of AI and healthcare. Over to you, Kexin.
All right. Hi, everyone. So it's really, so it's really lovely to meet you all. So basically, I want to ask that, have you ever wondered how old your body is compared to how old you are? So hi, everyone. I'm Kexin, and I'm a PhD student at the University of Oxford, and I specialize in computational biology. And today I'm going to present my bachelor's project in using the metabolites and DNA methylation to predict the age of our body. So before I get into the story, so what's my connection with EquiTech? So basically, after the second year of my undergraduate at Imperial College London, I joined EquiTech Features in a Scholars program, and shortly after, I started working on a project in the EquiTech Health Lab as a research associate. So we worked on predicting the stage of breast. So we worked on predicting the stage of breast cancer from biopsy images. And at the same time, I worked for 8 months on my bachelor's project to construct an aging clock. And these projects and EquiTech faculty helped me immensely to get a fully funded PhD via the Clarendon Scholarship at the University of Oxford, where I will be looking at irritable bowel disease. And honestly, I can't remember how many times Thomas and Abhilash helped me to polish my presentation for my PhD interviews. So today, I'm going to lead you onto a journey about predicting the age of our body. So first, why should we care about the age of the body? Well, as the quality of life increases, the number of years a person can live, or their lifespan, has increased much faster than the number of years they can live without any disease. So we now have this 9-year gap where people can be troubled by many chronic conditions. And in fact, as much as 80% of the American population are struggling with at least one chronic condition. So I have listed a few common ones that might be affecting you, your parents, or grandparents. So we might be thinking, can we prolong our health span to reduce this 9-year gap so that more people can live more healthily in their old age? So to do this, we first need to understand how aging occurs. So the process of aging is the deterioration of the body and its function over time. And the rate of decline is affected by lots of different factors. And depending on how each person is exposed to these factors, they can experience different rates of aging. So that gives rise to the concept of the chronological age versus the biological age. While the chronological age measures the number of years you have lived, the biological age measures the age of the body. So while both persons can be 40 years old, one who exercises regularly and eats a healthy diet can have the body of a 20-year-old, while the other who watches Netflix whole day in bed can have the body of a 60-year-old and live a shorter life. So we might ask, can we slow down the biological aging while someone is still young to improve their health in later life? So to do this, we first need to somehow measure the biological age. So scientists have been trying to use different measurements about the body to construct the prediction models to estimate the biological age. And we can call these models the aging clocks. So my research focuses on using DNA methylation and the metabolites to measure the biological age. But first, so what are the DNA methylation and metabolites? So DNA methylations are basically these markers on your DNA that regulate gene expression. So they act like switches to control if a gene is expressed or not. And the metabolites are substances that are made or used when the body breaks down food, drugs, or chemicals. They can be measured from the blood or urine and tell us about, for example, what a person has recently eaten. And these different measurements can tell us about different aspects of the body. And as a result, the aging clocks will also be linked to different aging risk factors. For example, a higher biological age that is predicted by the metabolites are linked to diabetes, being overweight, heavy drinking, and depression. And these are all somewhat related to metabolism. However, the metabolites are known to be very unstable because they can fluctuate with the food we eat. But DNA methylation patterns are quite stable as they're shaped by all the past environmental exposures and lifestyles. So imagine them as a record-keeping book in your DNA. So let's say you eat a donut every day, then this will leave a mark in your DNA that we can measure. So my research aims to use the DNA methylation as a marker to model the metabolic aging. So my hope is that DNA methylation can capture these long-term patterns in metabolites rather than the short-term fluctuations. So you cannot pretend to have a healthy diet by just eating an apple right before your blood test. It wouldn't keep the doctor away this time. So my hypothesis is that DNA methylation can be used as the metabolite markers in age prediction. And we can call these markers the DNA M metabolites. And next, we can develop aging clocks from the metabolites or the DNA M surrogate markers and show that DNA M surrogate markers are able to predict age more closely than the metabolites. And lastly, we need to assess these clocks against the risk factors to show that the DNA and metabolites are still sensitive to the metabolic aging risk factors. So to do this, I have been working with the airwave health study of around 800 people in the UK police force. So like any data scientist, I first split my data into training and test sets. And next, I use a machine learning method called penalized linear regression to select a subset of the metabolites to predict age. And finally, I developed the DNA methylation markers or DNA M metabolites for each of these metabolites, and I use these to predict the biological age. So how do we assess our model's performance? Well, we can see how well our predicted age, so we can see how they correlate with the chronological age in the test set. But what does the predicted age tell us about a person's health? So we can calculate age acceleration as the difference between the predicted and the chronological age. So basically a positive age acceleration would mean the person is aging faster than they should be. And then we can regress this on the different risk factors to find out how the different lifestyles contribute to accelerated aging. So Here you can see the age predictions from the two clocks plotted against the chronological age, with y equals x, meaning a perfect prediction of the chronological age. So essentially, we're saying that given a person whose age we don't know, we can make a very, very close guess of their age by just looking at their DNA methylations or metabolites. So we can see that DNA methylation surrogates can indeed predict age better than the metabolites from their stronger correlation with the chronological age, so R equals 0.91 here. But does it mean that DNA methylation surrogates can truly capture the risk factors of metabolic aging? Well, let's take a look at how age accelerations are associated with each of the risk factors. So as a reminder, age acceleration is the predicted minus actual age, or how many years your body is older than your passport age. So here the axis here shows the years of age acceleration associated with the risk factor relative to the reference level in brackets. So red is the age acceleration predicted purely from metabolites, and blue is the age acceleration predicted from the DNA metabolites. So here, if the error bar lies to the right of the zero reference line, it means that the risk factor is basically linked to faster aging. For example, apologies to the gentleman in the audience, but we found that males can on average age 0.5 years faster than females. And this also explained why men generally live a shorter life than women. And furthermore, we found that obesity and heavy drinking are linked to 1 to 1.5 years of faster aging. And in addition, smoking can also make your body 10 years older than your current age. But perhaps these associations don't really come as a surprise because we already know that obesity, drinking, and smoking are linked to aging. So more surprisingly, we found that mental health is very strongly linked to aging. For example, we see that PTSD or post-traumatic stress disorder and anxiety and depression are all linked to about 1 year of extra aging, even when they don't qualify for full clinical diagnosis, but just showing some minimal symptoms. And in terms of biology, this also makes sense because stress will be reflected in our metabolism, such as how we digest food and how we store fat, and these are all closely linked with the biological age. So, to summarize, I've shown that DNA methylations and metabolites can predict the biological age. And where the metabolite data are not available, DNA methylation can be used as markers for metabolites, and they can still capture the metabolic aging risk factors. And this is very useful because DNA methylation is more stable than metabolites and more standardized to measure. And more generally, the study illustrates how we can apply machine learning to biology to model the biological age. So, where can the project take us? Well, with an aging population and a lowered cost to measure DNA methylation, we can use the algorithm as a general screening tool on the population to identify those who are aging faster than they should. And using their biological age, we can make an early assessment of their risk for chronic disease years before they even get the disease, such as at what. So for example, we can predict anxiety and depression even before their diagnosis. And then we can advise these high-risk behaviors. Then we can advise these high-risk individuals of behavior change to lower their disease risk when they get older. And this can help us to reduce the public health burden and hopefully allow more people to have a healthier body when they get old. Thank you very much, and I'm happy to take any questions.